Hello, my name is Leah Jewel. I was assigned the KRAS protein with the mutation glycine amino acid number 12, which is replaced with cysteine G12C. The protein releases signals from outside the cell to the cell's nucleus. These signals instruct the cell to grow and divide and take on specialized functions. The KRAS protein acts as a GTPase, which means it converts the chemical GTP into another molecule called GDP. In this way, the KRAS protein functions like a switch that can be turned on and off by GTP and GDP molecules to transmit signals. The KRAS protein first binds to the GTP molecule. When the GTP is converted to GDP, the KRAS protein is deactivated, stopping it from sending signals to the cell's nucleus. Since it instructs the nucleus to do cell-specific tasks, I would classify this function as a receptor. The protein is present in both human and animal cells and is often located near the inner surface of the plasma membrane. Because the G12C mutation enhances the signal that regulates cell division and growth, the change associated with this mutation becomes more active. When this occurs, tumors may start to spread throughout the body, of course causing cancer. And now I'm going to show my 3D model here. So as you can see, it goes from outside the cell and then inside the cell, and then it signals to the nucleus, which is right here, and then it makes the cell divide. And on this side with the mutation, you can see here there's a very drastic change. When it's mutated, they send stronger signals to the nucleus, which is here, and the cell starts to divide uncontrollably, causing the cell to become cancerous. So yeah. The protein structure has changed due to the removal of mutations, resulting in more frantic signals and allowing a greater number of signals to travel through the protein. The primary functional groups found in the initial amino acid side chain is glycine and carboxyl groups. These two functional groups are nonpolar and hydrophobic. This amino acid is the smallest and the most basic among all amino acids. In addition to it being hydrophobic, the initial amino group may assist in forming bonds with other hydrophobic groups. Properties that are presented in the mutated side chain are amino groups, carboxyl groups, and sulfide hydrogens. The new mutated amino acid side chain is slightly polar. The polarity is pretty mild, so it's unable to properly interact with water, making it hydrophobic. The new mutated amino acids are smaller than the other essential amino acids. There are a few differences in comparison to the mutated amino acid in the original amino acid side chain. The mutated amino acid has a much stronger bond because the amino acid is much bigger, so it does with peptide bonds. The mutation affects the protein by sending more signals than needed through the cell, and it changes the shape of the protein as well. It affects the cell by allowing it to grow and divide at a rapid rate. This ultimately affects the organism because it causes a tumor, which of course leads to cancer. The main type of cancers that are associated with this protein are lung and pancreatic cancer. And for the limitations, I would say that with my model, I wasn't really able, or the model wasn't really able to show its active and inactive states for the protein, and it doesn't show the cell division or how the cell travels through the body and causes cancer. So yeah.